CRISPR, or clustered regularly interspaced short palindromic repeats, is revolutionary genome technology. CRISPR has resulted in major controversy both scientifically and socially, sparking heated debates. CRISPR is a man-made molecule that can detect mutated DNA. It utilizes a specialized enzyme called Cas9 that acts as a helicase and unzips the mutated section from the genome. If this happens in an egg, sperm, or embryonic cells, its change can be permanent and move on to future generations. The clustered regularly interspaced section of CRISPR refers to the portion of a genome that either synthetically edits a region doctors specify or destroys previously identified harmful injected DNA from outside bacterial sources. The history of CRISPR begins in 1987 with the discovery of repeated sequences of DNA in E. coli. Japanese scientist Yoshizumi Ishino and his colleagues published the sequence of a gene called IAP belonging to a gut microbe. As research into this newly found realm of repeated DNA sequences came to fruition, baffled scientist Francisco Mojica became the first person to realize that these seemingly identical strands of a genome actually shared common sets of features. After making this unique discovery, Mojica developed enough knowledge to coin the newly found sequences as CRISPR, a new frontier that would come to extend both advances in modern medicine and new controversy surrounding how far science could stretch its ethical limits. In 1998, an Arizona teenager, Jesse Gelsinger, became the first human to die as a result of gene editing therapy. He had suffered from an inherited liver complaint called OTC. Unfortunately, by injecting the virus that carried the gene to help his liver, Gelsinger's immune system rejected it, causing his liver, spleen, and other organs to be damaged. Gelsinger's death strengthened the controversy, as it raised the question as to how dangerous gene editing therapy actually is, and that more research may need to be done in order to be safe for human use. Two of the main scientists that were involved in CRISPR research, Jennifer Doudna and Emmanuel Charpentier, discovered that CRISPR can be used to edit human cells outside of the body. CRISPR is also a topic of high interest for medical researchers because it means that there is a possibility of treating heritable conditions such as cystic fibrosis and sickle cell disease, as well as DNA-infiltrating ones like HIV. In 2013, scientists found that CRISPR can be used to edit the genomes of everything from flies and plants to mice. Agricultural researchers were able to use CRISPR to modify various crops to be disease-resistant. Similarly, animal researchers recently performed research aimed at developing disease-resistant pigs, while working towards future goals of procuring a species containing humanized organs that are safe to transplant directly into a human's body. In November of 2015, British scientists successfully used the TALEN gene editing to cure a child's leukemia. TALEN gene editing is similar to CRISPR as tails, transcription activator-like effectors, are restriction enzymes that can cut the problematic sequence of the DNA strand, replace it with the correct sequence, and then let the cell do the rest of the work. TALEN was the first time a gene editing technology was used to treat a patient. However, the topic of gene editing creates several ethical questions among nations. In December of 2015, a 3-day human gene editing summit was held at the National Academy of Sciences in Washington, D.C., where it was decided that the U.S. would continue its prohibition on funding for gene editing research in human embryos, while China would allow it to continue. However, some companies experimenting with CRISPR, like CARIBOU Bioscience, don't believe in editing embryos, according to its CEO, Rachel Horowitz.
Broadly available.
Yeah, but I mean, we're not CRISPRing humans right now, is what you're saying. Yeah.
No, and in fact, our company has drawn a really hard line. We will never edit human embryos. We just full stop think that's a line that should not be crossed.
Right.
On May 24th of 2016, a French organization, Alliance Vita, launched their Stop GM Babies campaign in order to raise awareness about the ethical problems with genetically modifying human embryos and express their concerns about treating babies as guinea pigs, as well as the possibility of the procedure resulting in genetic mutations among babies. While CRISPR may be useful in saving people from fatal diseases, many argue against it on the grounds of moral reasons.
So, in a sense, it democratizes gene editing. And I have no idea where that will go. Are we wise enough to tame ourselves in the presence of this awesome power that previously was only in the hands of nature?
Many view CRISPR as a form of playing God, often based on their religious and moral beliefs. While this gene editing technology could be used to eliminate defects in embryos, there is also the possibility of using the technology to create future designer babies. If designer babies became a possibility, the idea is that those using the technology could give desired traits to their children, creating deeper class divisions as the rich who could afford the technology may create superior humans. Possible further implications of CRISPR, such as this, raise the question as to how far we should let gene editing go. Should it be unrestricted, used only to improve health problems, or not at all? In a poll conducted asking students about their views on CRISPR, 31.6% stated that the government should hold no such power. When asked if the respondents themselves would undergo genetic editing to ensure that their own offspring did not inherit a disease, 47.4% of our respondents stated that it would depend on the condition, and 26.3% responded that they would in order to protect their children. Following heightened ethical concerns surrounding the advent of gene therapy in embryos, a research team out of Sichuan University in China became the first group to actively attack a patient's immune system with the CRISPR-Cas9 system of aid. The team used a specially designed procedure to attack a characteristic gene of the person's lung cancer under which the Cas9 protein could remove the damaging gene entirely. Despite this newfound success, many citizens and bioethicists have no restraint in voicing their concern encompassing the rate of development and line of acceptability. Modern technological procedures have allowed an increasing number of sequestered treatment plans to become a reality, but at what cost do these sequences become too dangerous or unnatural for the human being? The line between medical necessity and personal desire in genome editing will always stand linked in an epic twirl of interrelatedness, a double helix of medical miracle aligned with progression towards a designer baby culture. CAR-T gene therapy, or chimeric antigen receptor T gene processing, is a medical system that allows doctors to successfully remove infected immune system cells, or T cells, from a patient's body. The CAR-T system is specially designed to target damaging bacteria and cancerous patterns in a cell's DNA. This specific procedure of testing became the first FDA-approved gene therapy program in United States history, aiming to relieve sufferers from a specific subset of leukemia. In August of 2017, a survey was conducted in which people were asked whether or not they support gene editing to treat medical conditions and restore health, and if they support gene editing to enhance and improve human abilities. The responders were organized into 3 categories: very religious, somewhat religious, and non-religious. From the graph, an interesting find was revealed. The highest number that supported both of those statements were people that identified themselves as non-religious, showing how a con-CRISPR view is likely usually tied to religion and morals. As of 2017, cancer is one of the main diseases that scientists are trying to find a permanent and harmless solution to. However, through the use of CRISPR, humans may be able to use FDA-approved gene therapy to treat localized cases of leukemia. CRISPR is a revolutionary piece of technology that offers tremendous opportunities for those suffering from cancer and various hereditary and genetic diseases. However, as Jennifer Doudna stated, CRISPR also offers the greatest peril for the future of humanity. For the past 30 years, it has been fiercely debated whether or not the human race should be given the power to play God. Is it ethically correct for mankind to define what is normal? Or would it lead to a synthetic outcome of designer babies and controlled breeding? The future is unclear. However, as science constantly pushes us forward into a technology-based society, progress and utilization of CRISPR will likely occur.